Autolysis, autophagy, and apoptosis. Autolysis, autophagy, and apoptosis are all cellular processes that involve the degradation or removal of cellular components, but they differ in their mechanisms, purposes, and contexts. Here's a breakdown of these processes, highlighting their key distinctions. Autolysis. Definition. Autolysis refers to the self-digestion or self-destruction of a cell via its own enzymes, typically lysosomal enzymes, following cell death or significant injury. Mechanism. After a cell dies or suffers severe damage, lysosomal membranes become permeable, causing hydrolytic enzymes to leak into the cytoplasm. These enzymes then degrade cellular structures indiscriminately. Purpose. Autolysis is a passive process that occurs after a cell has already died. It is not a programmed event like apoptosis or autophagy, but a byproduct of cellular breakdown. Context. Autolysis is commonly observed in necrotic tissue where cells die due to injury, infection, or lack of blood supply. Autolysis also contributes to tissue degradation after death. Outcome. The process leads to the breakdown and disintegration of cellular structures, often contributing to tissue necrosis and post-mortem tissue decay. Autophagy. Definition. Autophagy is an active, regulated process where cells degrade and recycle their own components using lysosomal machinery. It is a survival mechanism that helps maintain cellular balance and health. Mechanism. The process involves the formation of a double membrane vesicle called an autophagosome. This vesicle engulfs damaged organelles, misfolded proteins, or other cytoplasmic materials, which are then fused with the lysosome to form an autolysosome. The contents are broken down and recycled. Purpose. Autophagy is essential for cellular survival under stressful conditions such as nutrient deprivation, hypoxia, or infection. It helps remove dysfunctional components, thus maintaining cellular homeostasis. Context. Autophagy occurs regularly in healthy cells as part of normal maintenance, but it becomes more prominent during times of stress. Outcome. The process promotes cell survival by recycling cellular components and providing energy and building materials for new cellular structures. It also aids in pathogen defense and organelle turnover. Apoptosis. Definition. Apoptosis is a highly regulated and programmed form of cell death that serves as a crucial component of development, homeostasis, and immune defense. Unlike autolysis and autophagy, apoptosis is a deliberate process where the cell systematically dismantles itself without causing harm to surrounding tissues. Mechanism. Apoptosis is initiated by either 1, intrinsic pathways referring to internal stress or damage, or 2, extrinsic pathways referring to external signals. This triggers a cascade of molecular events involving enzymes called caspases, which break down cellular components in an orderly manner. The cell's contents are packaged into membrane-bound vesicles called apoptotic bodies, which are then safely engulfed by neighboring cells. Purpose. The primary role of apoptosis is to remove damaged, infected, or unnecessary cells without causing inflammation or damage to nearby tissues. It is a controlled beneficial process that helps in growth, immune function, and tissue renewal. Context. Apoptosis occurs in many situations, including during development, for example, shaping of fingers and toes, in response to DNA damage, or to eliminate infected or cancerous cells. Outcome. Apoptosis ensures clean removal of cells, maintaining tissue health and preventing damage or disease. When dysregulated, it can lead to conditions like cancer, too little apoptosis, or degenerative diseases, too much apoptosis. Key differences. Initiation. Autolysis, triggered by cell death or severe injury leading to uncontrolled enzyme release. Autophagy, a regulated process initiated by the cell to cope with stress, damage, or nutrient deficiency. Apoptosis, programmed cell death initiated by specific signals, either intrinsic or extrinsic. Autolysis occurs after cell death, contributing to breakdown and decomposition. Autophagy helps maintain cellular health and survival by recycling cellular components. Apoptosis safely removes damaged or unnecessary cells to protect surrounding tissues and maintain balance. Autolysis involves the release of lysosomal enzymes into the cytoplasm after cell death. Autophagy involves the formation of autophagosomes that transport cellular materials to lysosomes for degradation. Apoptosis involves caspase activation, controlled breakdown of cellular components, and formation of apoptotic bodies. Autolysis results in cell disintegration and tissue necrosis. Autophagy promotes cell survival by recycling cellular materials. Apoptosis results in orderly cell death, removing unwanted cells without causing inflammation or tissue damage. Each process of autolysis, autophagy, and apoptosis has distinct roles and outcomes, from passive cell disintegration in autolysis to the survival-oriented recycling of autophagy and the clean, programmed removal of cells in apoptosis. Together, these processes illustrate the complexity of cellular life and death, and their regulation is key to maintaining overall organismal health.